Okay, so this is the fourth class in our human cell engineering course. This is the one that's going to be the most interesting. This is the one where we actually get to genetically modify the cells. So last week you should have run an antibiotic screen testing your cells to see what their tolerance for G418 antibiotic was. And your answer should be somewhere between 200, maybe as much as 800, but probably somewhere around 400, 600. And what you're looking for is the maximum amount or the minimum amount that will kill all the cells. So after a few days, you should see well after well of cells dying, and then you should see some cells that have very little that didn't die. And if those wells still have happy thriving cells, then go up one set of concentration to the lowest number where they all died. The reason you want to do that is the plasmid we're going to be adding today confers two genes. So it confers the LAC-Z gene, which is what's going to help us. It's actually our gene of interest, but another gene which makes these cells resistant to the G418 antibiotic. So we want a low dose because it's resistance. They're not immune to the effects of this antibiotic, so you don't want to just massacre them with huge amounts of antibiotic, but they will have a better chance of surviving than cells that are that don't pick it up. So the idea is your cells, after they pick up the DNA, it won't be 100%. Some number of your cells will pick up the new DNA. Then you'll add the G418 antibiotic, and that'll kill off any of the cells that don't have it. That'll allow the cells that do to continue to propagate and to sort of spread their new genes, and that'll make more cells that do have that antibiotic resistance gene and our gene of interest. So the process is pretty straightforward. The number one thing is work really clean. Contamination's your enemy, and I'm sure if you've gotten this far, you've probably seen a little bit of contamination. We do have antibiotics and antimycotics, which is like antifungals, in the media that we've sent with the kit, but still stuff can grow in this, so be very clean. First thing, I took my workbench and I cleaned off the area with like a Lysol wipe kind of thing, just to sort of get rid of any big debris. And then I'm going to take some alcohol and spray the space down. I like to put a paper towel down. That way, anywhere I need to put something, lay a lid, anything like that, I can put it on the surface. It's not just sanitized, but sanitizing. And then clean your hands up. So it's important every step along the way. If you handle something, you go pick up something that you haven't used before, you open a door, come back, clean your gloves, clean your arms. I usually go all the way up to my elbows. I'm going to be working a little unsanitary today because I'm just demonstrating, but wear like a surgical mask, things like that, because your breath's full of bacteria and you're going to breathe it in those cultures, and some of them may be able to survive even with 3 different antibiotics in the culture. So what we want to do, first thing, you're going to see this LAC-Z plasmid, right? Well, I don't know if it'll focus on this, but LAC-Z plasmid. It's freeze-dried. So there's DNA in here, and it should just look like kind of a little smear at the bottom. It may not look like much at all, depending on how freeze-dried it is, but we need to resuspend that. So the first thing we're going to do is resuspend that in 55 microliters of water. You should also have received a little tube that says sterile water. So I'm going to set our pipette to 55. You should be wearing a mask this whole time. I'm not going to, just so you can still hear me. Join 55 microliters of water. Add it to the LAC-Z plasmid, because plasmid's been freeze-dried, all of its water's been removed, and so it's just DNA in a tube. Now it's DNA and water in a tube. Shake it up thoroughly. Sometimes when these things are shipped, the little clump of DNA will get knocked loose and it'll be in the cap or something like that. So, just try and kind of keep your eye on where it actually is. But give it a good shake. And that should give you a nice resuspended plasmid. Okay. So So now your plasmid's been resuspended. First thing you're going to do, and I recommend doing that first so it's just got plenty of time to get fully resuspended. First thing you do is change the media. So you should have a 12-volt plate that looks like this that you did your antibiotic screen in. You can do exactly the same thing you did last week to make a new plate. You can take another flask, you can take those cells, do exactly what you did before, and make a new plate. So, exactly the same protocol, fresh plate, and you're going to do exactly what you did last time. You're going to continue to grow them up, you're going to watch them under your microscope, and once they reach about 70% confluency, so once 70% of the floor, of the bottom surface of the well is covered in cells, so you don't want 100%, you want lots of cells, but you want room for them to continue to grow. So about 70% is ideal. If it's a little past it or a little before, it's okay, but you'll get your best results if you time it right. So you want about 70% confluent cells. The first thing you can do is replace the media. So that's pretty straightforward. You can do this with your mask on. You set your bigger pipette to 1,000. You take your media. Y'all's media is going to be in a bottle. And just be careful, don't contaminate your media. Let's see. You'll need something to dispense your waste into. This is just a bottle with a little bleach water, but it can honestly be anything. None of this stuff is, dangerous. It's not like human cells are going to get out and start growing out in the wild. Okay. So first thing you want to do, prep that. You're going to prep 6 wells. So try to find 6 wells that are good representatives of 70% confluency. Nice healthy wells. So what you're going to do is just try to do this in a way that's less likely to be contaminated than I'm doing it. I recommend just sort of drawing it off, dispose, and then immediately refilling. You don't want them to go too long without media, and I don't recommend having it open like this. I'm having it open so you can kind of see, but I usually just tip the cover and then draw the media out. That way things in the room, things off my arm don't just fall in it. Try not to pass your arm over exposed wells because things will just fall off of you into it, spores, bacteria, all kinds of stuff, and it'll contaminate everything. But you're just going to draw 6 wells, and you're going to replace their media. It's important they have fresh media. And you're going to label those wells so you know which wells you're working with. You can replace all of them if you want to, but it just depends on how much media you've got left and how many plates you've had to do. Sometimes things don't go smoothly. You have to do steps over and over again, and you wind up with different amounts of media. So you're going to label 2 as your controls, and these will not receive any DNA, and you're going to label 2 as your minus DNA, and 2 that are going to get the DNA. So it's going to be a negative control, and then no DNA, and then no DNA, and then so you label your wells. It's important to keep track of which wells are which because that's going to help you interpret the results. So what you're going to do, you're going to take you take 4 microcentrifuge tubes, right? That's going to be these little tubes. You're going to put them in your tube rack. You see that? Okay. So these four are going to go in your tube rack. And this is where we're going to build our reaction. So we're going to be using calfectin and our plasmid today. So calfectin is a calcium-based transfection reagent. What it does is it uses calcium ions that are positively charged to neutralize the natural negative charge that DNA has. And that helps it to pass through the negatively charged cell membranes by just neutralizing those charges so that the DNA and the cell membrane don't repel each other so much. So this is a pretty old and well-established method of genetic modification, but it works really well. So we're going to build our reaction. So you can take more of your L15 media, and you're going to put 50 microliters of each. So we'll set this to 50. I'm going to put 50 microliters in each microcentrifuge tube. 1, 2, 3, 4. And again, be really sterile. Don't just breathe on your media like I just did. Wear a mask. Okay. So you should have a very small amount, not very much at all. Kind of just a drip's worth. Okay. So we're going to go in 2 tubes. Let's go ahead and label them so that we don't get confused. It's one of the very important things when you're doing this kind of stuff is that you label everything, because you don't want to get halfway through pipetting stuff out and you get a phone call or a stray thought distracts you, and next thing you know, you forget which tubes have DNA in them, which tubes have calfectin in them. You don't know where you are. You throw them away and start over. Labeling saves you. So 2 are going to be minus DNA, and 2 are going to be plus DNA. Okay, plus DNA. This is going to give us a basis for comparison at the end. So these 2, so all of them are going to get calfectin. 2 of them are going to get calfectin and DNA. So you're going to separate them into 2 little groups. The calfectin is, actually we're going to add the DNA first. So we're going to go DNA first at 10 microliters. So set this guy to 10. Okay. You can take your LAC-Z DNA. And the LAC-Z gene encodes for an enzyme called beta-lactamase. Beta-lactamase is used in, or excuse me, that's a different gene, galactosidase. Beta-lactamase is one I'm much more used to dealing with, but yeah, so LAC-Z encodes for galactosidase, beta-galactosidase. So what it does is it's used to break down, it's used by E. coli to break down galactose. And galactose is a sugar. But we're kind of hijacking it with this. So, all right, we've got 10 microliters of our LAC-Z. So we're going to go 3 microliters now. So 3 microliters, it's hot in here, of calfectin. And all 4 of these will get 3 microliters of calfectin. You won't have a lot of calfectin. It's the most expensive reagent in the kit, so be careful not to spill it. And one thing I like to do is just, you know, when I add something to it, I'll put it on a different row. That way, if I get distracted, I know that the ones that are on this row have already received their, you know, whatever it is I'm dispensing, and the ones on the front row have not. And that way, you can just keep track of things. So we're going to do all 4. This way, at the end, we'll be able to compare whether the calfectin, we'll be able to see the effects of the calfectin versus no calfectin, and we'll be able to see the effects of calfectin plus DNA versus none at all. So, alrighty, and there we go. All 4 now have their calfectin, and these 2 have DNA, and they've been labeled. So now we're going to need to wait about 10 minutes. So set a timer, wait 10 minutes before you do the next step. I'm not going to wait, but I am going to explain for a second while mine works. What's happening now is the calcium ions in the calfectin are just having time to float around and run into DNA and bind to it. So you have to give this reaction a little bit of time, just so that they have a chance in this sort of concentrated form before it gets diluted in all this much larger amount of fluid. It has this chance to really find each other and bump into each other. Because once it's diluted, it's going to be much longer before they can find each other. So you want the DNA to be very neutral before you add it, and then it should go through and do its job. Now, cell membranes don't like to be neutral. Proteins don't like to be neutral. DNA doesn't like to be neutral. There's a lot of things. Transfection reagents, generally speaking, are toxic, cytotoxic, so toxic to cells, because cells just generally don't like having DNA from the outside stuck into them. So they resist this. And so the things that you have to do to them to make them uptake DNA from the outside are pretty aggressive. And in this case, they'll take up the DNA, but the cost is, it's harmful to your cells. So that's why we're only going to leave it sitting on there for about 24 hours before we change the media again. Now, it's been 10 minutes, we'll say. You're going to take your wells, and you're going to have them labeled. 2 are going to have DNA, 2 are going to have no DNA, 2 are going to get nothing at all. So all they're going to do is have, they're going to be growing in the same conditions, and they're going to have had their media changed. These 2 are going to get calfectin and media. These 2 are going to get calfectin, media, and DNA. And all you're going to do is just add that in. So, go about 60, 65, so you have plenty. And just pipette this out. And this is one of the ones with DNA. So I'm going to add that. This is one of the ones with the DNA. It's better to change pipette tips between these things. I'm just trying to be quick for the sake of the video. Take your time. Alrighty. The next 2 are going to go into the one that says no DNA. So double check your labels. No DNA. And no DNA. Now, this is going to give us controls so that we can evaluate the experiment. If everything goes right, your controls will just be something to contrast against. If things go wrong, they'll help you to diagnose what went wrong and help you to get it right on the next time. So, at this point, the day that you do this, you put them back in the whatever you got set up, your incubator, your heating mat, whatever it is. You put them back in the nice warm conditions so they can just sit there and do their thing. Tomorrow, about 24 hours later, so about the same time of day, so plan ahead, make sure you have time tomorrow to do these things. You're going to change the media, and that's going to be all 6 wells. So we're going to do exactly the same thing we did before. Get your big pipette, set it to 1,000. You're going to pull off all the media. And this has been sitting in here for 24 hours. Remember, I'm doing this for the video now, but give it a full 24 hours. that will give the DNA time to get into the cells, and that'll give them time to, grow up some. So you'll have a few cells that have been genetically modified, and they'll be just at the beginning of starting to express some of that DNA maybe. It takes a couple of days. The very, very beginning might be in about 24 hours, but usually you get stronger expression after about 48. Now these cells have been through a lot. They've had their media changed. They've been through a couple of different temperature changes. They've had transfection reagents and new DNA and all sorts of stuff added to them, so they're not very happy right now. So we need to give them a couple of days to recover. So this is the next day. We transformed them yesterday. We changed the media today. Back in their incubator, warm space, and just check on them. So put them back on the heat, back in the warmth. Give them a full 2 days. So 48 hours from now, you'll proceed to the next step. So during this time, you may check them under the microscope and they look pretty rough. And that's normal. So because of the stress of the transfection reagent, it's harmful to them. Like I said, it's cytotoxic. So your cells may look pretty bad. It's okay. As long as they're not all dead, floating around, and it's just debris, you're okay. As long as there's cells and they just look stressed, okay, put them back. They got fresh media. You got rid of the transfection reagents and the DNA. The only stuff that should be left is what's inside the cells. You give them nice fresh media. You give them a couple of days of warmth and comfort to recover. So 2 days later, they've recovered. Now we're going to stress them again. Now we're going to add the G418, and we're going to add xGal. Now the amount of G418 that you add depends on the test that you did last week. So if you found 400 microliters or 400 milligrams per milliliter was the lethal dose, or like the ideal dose for your cultures, then use that same amount. So that will tell you if it's, these come pre-diluted to 200 milligrams per milliliter. So if 200 milligrams per milliliter is that tells you that's how many micrograms you're going to use basically. So if 400 was your target, you're going to use 2 microliters. If 200 was your target, you're going to use 1 microliter. If 600, you're going to use 3 microliters. So you set your pipette, dispense that, and your xGal solution. So in my case, I'm going to go with 2. That's what I usually find is it's about 400 microliters of, or 400 micrograms. So this is 1,000 times concentrated. So since it's 400 and this is 200, I'm going to use 2 microliters to make 400. So I'm just going to add the G418. Now it may go ahead and start changing colors a little bit as soon as you add these next 2 ingredients. That's okay. The reason it's pink, the media that we send, is because it has a pH indicator. You may have seen some of your flasks and things turn yellow. That just means that the pH has shifted. This stuff will turn, the dye will turn yellow if it gets into an acidic pH, and it'll stay pink if it's in like a happy pH. So you're going to add that into all 6. So that includes the ones where we didn't add DNA or calfectin, right? So that's your G418. Now that means what we should expect is that these 2 that got their media changed but didn't get any DNA should die from the G418, just like the ones in your antibiotic screen did. The ones that got calfectin and didn't get DNA should also die. The ones that got the DNA should live because they're resistant to G418. But on top of that, we've got our xGal solution. So XGAL is a sort of synthetic chemical that's similar to galactose. So remember I told you that the LAC-Z gene encodes for beta-galactosidase. So beta-galactosidase will cleave a galactose. So it's part of how bacteria digest galactose, which is a sugar. So it's, when they're digesting it, they cleave it into smaller molecules and break it down. Well, X-galactose is XGAL is a synthetic chemical that has basically a blue chemical bound to a sugar in such a way that when beta-galactosidase digests it, the blue becomes blue. So that means this stuff, even though it looks clear now, and it should look clear, if it gets old it can turn kind of green, but it should look clear. If it doesn't, we can send you new stuff. But this stuff is you know, colorless for the most part in terms of the quantities we're looking at, and when it digests it should be a bright, vivid, distinct color. So it will turn blue as the enzyme digests it. So the enzyme was encoded in the DNA that we included, so only those cells with the DNA should have made that enzyme over the last couple of days and should be able to digest this stuff and turn it blue. That will be your really clear sign that things have worked. And you see how it's turned a little bit slightly different, a little bit yellowish. That's just because the pH is a little different in the G418, and it will, you know, kind of swing things a bit, but not enough to be a problem. If it's not like orange or yellow, the cells will be fine. All right, so you're going to add your XGAL solution, and let me check my notes just so so add about 15 microliters of XGAL. Let's see. So pick up your pipette. You know, and the sensitivity will depend on, you know, how many cells you have, how well those cells have been modified. You know, if you're very successful, so start with your DNA ones, add the XGAL solution. Make sure you keep track of it. Should have just enough to do the last one. Okay. So all 6 get the XGAL solution. Now, what you should see over the next few days is, if everything worked perfectly, these wells will die. These untouched wells will live. So they have just been growing the whole time. You didn't change media, you didn't touch them at all. They should live. They might become densely overgrown, but they should live, and there may be some cells dying, but as long as they don't start turning yellow or dry out or something like that, they should live. If they do, you can feel free to change their media. The top 2, so the 2 that just got their media changed on the first day, didn't get any DNA, didn't get calfectin, didn't get anything on the intermediate steps, and just now got G418 and XGAL, should die. These 2, the 2 that got the calfectin but did not get DNA, shouldn't have the genes to turn anything blue, and they shouldn't have the genes to resist the effects of the G418 and should die. These 2, the ones that got the DNA, got the calfectin, got the whole process, have been genetically engineered. Now they have 2 additional genes. They have resistance to G418, so they should survive. They also have the LAC-Z gene, which allows them to make beta-galactosidase, and they should be able to break the XGAL down into a blue compound, and the whole well should turn bright blue. It should should look basically like this, right? So they may look a little more purple. If your well is a little overgrown, so there's not a lot of room for new cells to grow, you'll end up in a situation where you have less expression because you have fewer cells that have been modified because you haven't had a chance for them to reproduce their modification. So you'll get more of a purple somewhere between the sort of pink and blue. If you get if your cells are very few cells, like if your confluency is low, say 30 or 40%, then you'll have very few cells to start with, and everything will be slower. Or if you do it too early, or if you use less than 15 or whatever, you may end up with, or just generally your transformation wasn't very successful, or your cells are stressed, you may see a more purpley well than a bright blue one. If everything goes really, really well, you see nice bright blue wells like these. And anything that has DNA added, and you may have to jumble it up, right? So if you're having to find happy wells, you may have to your positive controls over here and other controls are over there. So but what you should see is the two that have been modified should be very clear and distinctly bright, beautiful blue. The rest of the cells that you didn't touch at all should live and stay pink. Maybe turn a little yellow or orange if they're starting to get a little old. And the wells that have been treated with everything but the DNA should die. So that will give you, and this will take, if you've done the full thing and you keep them on the incubator, you may start seeing something within hours, but you should see something within a day or two. If you don't see something within a day or two, it's time to repeat the experiment.